Hello, everybody. So I'm not going to repeat the title of the presentation. I'm going to go directly into what we're going to talk today, which is grapevines. The grapevines belong to the genus Vitis, which has about 60 species. They are perennials and vegetatively propagated. There are about 30 species in North America and another 30 species in Asia, but the most widely cultivated species is the Vitis vinifera, which was domesticated about 6,000 to 10,000 years ago. So just an acceleration, when I'm talking about hybrids here, in grapevines, when you do interspecific crosses between Vitis vinifera and wild species, they're usually called interspecific hybrids or just hybrids. So when I'm referring to hybrids here, don't think on maize hybrids. It's something completely different. Why interspecific hybrids? So grapevines, Vitis vinifera has an excellent fruit quality. I hope you all have tasted in wine, table grapes, or raisins. But had poor tolerance to biotic and abiotic stresses such as diseases, drought, and cold. On the other hand, there is a huge array of wild relatives, and some of them have interesting traits like disease resistance, pest tolerance, cold tolerance, but they haven't been selected for their quality. So they have poor fruit quality, small fruits, thick skin, big seeds, and also of flavors that stay in the wine and are not desirable at all. So the motivation for the program and the motivation for this research is that the integration of disease resistance and other favorable traits into cultivated Vitis vinifera will allow the development of new grape varieties with less fungicide requirements and better tolerance to adverse conditions. So in order to do this, we need to understand the grapevine genetics. Grapevines are diploid, highly heterozygous, with a rapid decay of the linkage disequilibrium. And while the genus is highly diverse, most of the Vitis vinifera varieties that we consume are derived from a few elite parents, so there is a lot of room for improvement here. They have a relative medium to small size genome with 19 pairs of chromosomes. And most of the genetic studies for grapevines are QTL mapping in bi-parental F1 families using the pseudo-test cross strategy. In this strategy, what you do is you use markers. Here I have a small scheme that segregates either on the female parent or in the male parent. So you look for markers that are homozygous in one parent and heterozygous in the other, and then you construct a female parental map and a male parental map. The objectives for this research in this thesis, I'm going to show some of the topics that I researched. First was the generation of high-density genetic maps from genotyping-by-sequencing SNPs for 4 Vitis gen F1 families. The problem with basically with the genotyping-by-sequencing and heterozygous is you have a heterozygous undercall, and then you have a lot of problem with the missing data that a lot of you know already. Then the second objective was to identify QTLs controlling the abundance of predatory mites, trichome, and domatia architecture in grapevine leaves. And the third objective was to characterize and identify loci linked to resistance or susceptibility to Phomopsis, cane, and berry spot in crosses involving Vitis cinerea. So going for the first objective, we're happy that this is already published. It was published last month. I work with Vitis gen. This is one of the main objectives of the Vitis gen project, and this was a work that was led by Katie Haima, where she developed a pipeline for generating genetic maps out of heterozygous crosses. The crosses and phenotypes that we use in this work, I'm going to talk about 4 crosses. They are Erysiphe F1 families with 5 common parents. I'm going to explain a little bit here. We have some crosses with Vitis rupestris by Horizon, Vitis vinifera Chardonnay with Vitis cinerea, Horizon by cinerea, Horizon by rupestris, and Horizon by Illinois. We use all these 4 populations to create genetic maps, and then we compare the maps for the different parental maps created from different crosses. And we validate this with the genetic maps were validated using the sex locus, which is a single dominant locus with 3 alleles, which is very well characterized in grapevines. So I'm going to go fast into the pipeline because I just don't want to spend too much time explaining the pipeline, but just a quick view. We start with the GBS SNP call. After you have your SNPs, you do a population level quality control, basically to remove any individual that is or a self or an outcrossing, because that will affect the results later. And then the first biggest step is the identification of pseudo-test cross markers and genotyping error correction. For this, we use Perl-based scripts that Katie developed for here. And here are some of the results. I'm just showing here in each pie chart is each of the families that we work, and I just wanted to show where the SNPs goes in this stage. Most of the SNPs are removed because they have low genotyping rate, or once you remove the error, they become with low genotyping rate. Blue, red, and purple are the markers that are removed for this reason. There is, for each, we started with around 450,000 markers in some of the population, up to 300,000 markers. And what you retain at the end is about 4% of the markers. There is between 10 and 17% of the markers that had incorrect segregation patterns. So as explained before, we're using a pseudo-test cross strategy. So we're looking markers that are homozygous in one parent, heterozygous in the other, which means that they have a minor allele frequency of 0.25. There is a huge proportion of markers that don't have this frequency, and that might be markers that are heterozygous in both parents, or 3 allelic, 4 allelic, or just bad markers. Those are removed in this stage. The next step is to create the linkage groups, and this is done by a pipeline that was developed using the R package WGCNA. And basically, what we do is to a hierarchical clustering of the markers topological overlap matrix, which in a figure is look at a dendrogram that is cut as some height that is defined by the user, and then it creates your linkage groups. The linkage groups after that are joined because sometimes they got separated, and we determine the phase and the marker order. For this we use MST-map, and then it's map curation. What we use was RQTL for this. Again, the markers by stage. Each pie chart, it's a population, and in this case you see that the distribution of the markers is more related with the size of the population. For this one we have 366 individuals, is the biggest one, and there is a lower proportion of duplicated SNPs because you have more recombination, and also less markers were filtered at the LG assignment. This population, which is the bigger one, is the one that retains more markers in each map, and about 7% of the markers in each of the 3 populations were removed during the curation step. The genetic map curation was necessary because after the ordering, we get linkage groups that were about 10 times or even more larger than expected, which is a typical problem that you have with GBS data. It's just because you have bad markers in your linkage group that makes your map just to go higher. So it's a little bit of a pain because it's kind of a manual curation for this. But after we did the curation for all these maps, we compared the results. One of the questions was if these markers were located in regions that were repeated regions of the genome that are not that don't look repeated in the reference genome. So if the markers, in order to assess if the markers are located in repeated regions, you cannot look at the minor allele frequency. You need to look at the minor tag frequency. So you need to go to your BCF file and look at the proportion of the tags. In the SNP that stay in the final map, you see here the minor allele frequency and the minor tag frequency, and you have a peak at the same 0.25, which is about 1 to 3 segregation. And you also see a small peak here for the minor tag frequency that is not present in the minor allele frequency in 1.7, which is duplicated regions. But if you look at the SNPs that were removed, for the same SNP, the pattern of minor tag frequency and minor allele frequency are completely different. For the minor tag frequency, you see that the values are below 0.1, which means that these tags are located in repeated regions. It suggests that these tags are located in repeated regions. which is kind of interesting to see if we can use this information in order to make the curation of the maps a little bit more faster. So these are the results. These are the genetic maps for the horizon parent. I'm just going to show for one of the parents. This is the 19 chromosomes. In these plots, you have the physical distance in the x-axis and the y and the genetic distance in the y-axis. All the different colors are independent maps. We have 3 populations with 2 pipelines, the de novo and the synteny pipelines. So these 6 independent maps. And as you can see, the maps were very well correlated. They look pretty much similar. In some cases, a little bit shorter. The x-axis go to the wall size of the reference genome. So we have genetic maps that cover from extreme to extreme, which is pretty good. The total genetic distance in the final maps were around 1,200 centimorgans for an average of 1,700 SNPs in the synteny pipeline. And in the de novo pipeline, we were able to keep more markers, an average of 3,200 SNPs with 1,300 centimorgans for these maps. So we thought these maps were good enough to QTL mapping or to map a single loci. What we used was to map the flower sex locus, which has been characterized and is well known where it is located. So in the upper part of the figure, you have the physical map for chromosome 2, and in the lower part, there is 3 genetic maps that we created. So this is the region where the sex locus has been identified, and we were able to map in the 3 genetic maps exactly in the same region for these 3 populations. The other thing that was interesting to see, these lines that connect the physical map with the genetic map is the position of the SNPs. And based on the genetics, we thought that these scaffolds that were originally not in the reference genome in the chromosome 2, we thought that these scaffolds should be added to this chromosome. And during the time of our research, a new version of the reference genome was released, and we found out that the scaffolds that we predicted that should be added there now are there. So that was a nice proof that our maps are doing a good job. So as a summary, the headmaps pipeline solves the problem of handling large number of markers and provide automated filters based on the SNP quality rather than physical position or parental genotype, which is what most people in grapevines is doing right now. The number of markers in the final maps is highly correlated with the number of individuals in the family, which makes sense because it's based on average recombinations. The genetic maps were consistent between pipelines, as well as for the common parent in half-seed families. And the analysis of the sex locus demonstrates the power of our approach, leveraging linkage, sequencing, and alignment to the reference genome to quickly characterize locus. So that was one of the first works that we did. The second objective was we worked with a quantitative trait to identify a QTL controlling the abundance of predatory mites, trichome, and domatia architecture in grapevine leaves. So it is well known that there is a positive correlation between the trichome and the predatory mite abundance in grapes. The trichomes are the hairs on the. If you've been in the repository in Geneva, you have seen that there are some of leaves that are very hairy. These are the hairs. The other trait is the bristles, which is this short, upright hairs. And there is also this trait called the domatia, the acarodomatia, which is a kind of a tough pocket in the axil veins, where it has some. It looks like a group of bristles that get together, and the entomologists have found that the acarodomatia like that pocket to put their eggs or to find shelter from predators. So all these 3 traits is being studied that is correlated with the abundance of predatory mites in different genotypes of grapevines. And the motivation for this in long term is to breed varieties that are able to sustain predatory mites in order to look for biological control. If you add these predatory mites, into grapes that don't have any hair on the leaves, the mites are lost after a while, so you cannot use it for biological control. The trichome pathway is well characterized in Arabidopsis thaliana, but it's not that well characterized in grapevines. Basically, I'm going to explain this very short. You have the GL2 protein, which is required for the trichome initiation. This protein can be activated for this activator complex that have these three components. There is also a negative regulation from this set of proteins. I'm not going to read it just for a matter of time. And then there is gibberellic acid dependent negative regulators, which are more related with differentiation, which are these proteins that are here and really well characterized in grapes. In Arabidopsis thaliana. So the work that we did here was, the working title for this paper is Genetic Locus Associated with Predatory Mite Abundance Co-segregates with loci controlling trichome and domatia density in grapevines. We used just one of the populations, the Horizon by Illinois cross, and the phenotype that we used was to count the predatory mites. For domatia, we look at the size and the density of the bristles using a categorical scale. And for the bristles and hairs, we also use a categorical scale of density. This is how the parents look like. This is Horizon, which is at just one hair here and almost no bristles. And this is the Illinois parent with the bristles here and the hairs along the veins. So the first result, we kind of corroborate what is known in the literature. The Phytoseiid abundance, which is here, is positively correlated with trichome abundance among the progeny. Here you see only white and red because we didn't find any negative correlations. We scored this for 3 or 4 years. We found some year by year effect. But overall, the Phytoseiid abundance is correlated with the traits. Of course, the traits seems to be more correlated with things that are related, like bristle in veins with bristles in blade, or the abundance of hairs in blades with the abundance of hair in the veins. This is the distribution of the phenotypes. We have an average count of phytoseids of between 0 to 10 phytoseids per leaf in the progeny. And the distribution of the bristle, hair, and domatia rating were across the scale between 0 and 9. So this is the result for the QTL mapping. We found several QTLs for this. We had mostly additive models for this. What I want to highlight first, we find a QTL for Phytoseiid abundance in chromosome 1 of the Illinois parents. And which is interesting because it's a different trophic level. For the entomology, it's very interesting. And then we found that this QTL also co-segregates with QTLs for all the other traits. So there is QTLs for domatia size, for bristle in veins and blades, and hair in veins and blades. This QTL in chromosome 1 was the one that explained the higher proportion of the variance for all the traits, and also usually was the one that had the higher LOD. We also found different sets of QTLs for the traits, and which is also interesting. We found that, for example, in hairs, there are different sets of QTLs for hair blade than QTLs for hair located on the blade and hairs located on the vein. Just here a table just to show you a little bit how is the architecture of the traits. The bristles was the trait that the simplest trait of it, and with what QTL you are able to explain about 55% of the variance. The domatia size was the second simplest with 2 QTLs in chromosome 1 and chromosome 15. These QTLs are the same QTLs that you found in bristles, which kind of makes sense because the domatia is composed of bristles. And then in hairs, you find QTLs, again, the same QTL in chromosome 1, and then different sets of QTLs for veins and QTLs for blades. For the Phytoseiid counts, which is the predatory mites, we found 1 QTL, and the percentage of the variance explained was variable depending on the year, and which makes sense because they're more prone to year-by-year variation. We look for candidate genes, and the regions, the interval regions were kind of large, so it's kind of hard to say that this is the gene that controls, but anyway, we look for candidate genes, and what I would like to highlight here maybe is for chromosome 5 and 8, which were these chromosomes, these QTLs for hair in vein or in blades. We found these proteins that are known to be are gibberellic acid dependent and are known to be involved in differentiation of tissue. So this is kind of an interesting candidate for this. So I left this research until this point. We found the interesting thing, we found markers associated to this Phytoseiid abundance QTL. We located this QTL, and it had an effect of about 3 Phytoseiid per leaves. And it's an interesting topic for research and interesting question for the breeders if they will be interested to breed for a QTL that it gives you 3 more mites per leaves or not. The architecture of the trichome trait with this predatory mite QTL having a major effect on all the other traits might explain the correlation observed between Phytoseiid abundance and leaf trichome in previous ecological studies, and also suggests that may be one or several genes involved in the general leaf development pathway controlling this trait here. Also, the additive effect of the QTLs suggests that stacking alleles or loci with minor effects could increase the number of Phytoseiid per leaves in order to sustain higher population of predatory mites, which is what is needed for effective biological control. And I might be talking too fast because, well, I guess that's okay for you. I'm going to talk to the third objective. Maybe I'll try to talk slower now because I rushed through all the slides, which I used to do. So the third work was to characterize and identify loci linked to resistance or susceptibility to Phomopsis, cane, and berry spot in crosses involving berry cinerea benign. So the Phomopsis cane and leaf spot is caused by the hemibiotrophic fungus Diaporthe ampelina, which was previously called Phomopsis viricola. That's what's the name of the disease come from. This is a monocyclic disease, and it overwinters in dead wood. So it doesn't create an epidemic like powdery mildew or other fungal diseases, but it stays over time. And since the grapevines are perennials and you expect to have the vineyards productive for 20 years, it's a disease that in the long term can cause great losses. It also can reduce the productivity when it affects the shoots, they break. Also, it affects the rachis, and the rachis is the skeleton of the cluster, and it gets dry, and then the berries fall to the ground. And also sometimes the berries get sour, and that can cause that the juice loads can be rejected because of this disease. This can be controlled by pruning and sanitation, but of course pruning and sanitation has demand a lot of labor, so it's something that the growers will be interesting to get rid of and to have some resistance. So for this work, we use 3 families. We have the Chardonnay by cinerea cross, Horizon by cinerea cross, and Horizon by Illinois cross. Now, Illinois is a progeny of Vitis rupestris B38 and Vitis cinerea benign. So basically, Vitis cinerea is represented in 3 of the populations of the families, and Horizon in 2 of those. So this is the phenotype that we observe in the field. This is a score 0. There is no Phomopsis symptoms found. The cane looks really well. This is a small infection, a small number of individual lesions. For the score 2, this is a moderate infection when the lesions start to coalesce and spread in the base of the cane. And this number 3 is a is a vine that is pretty well infected. The infection is severe. The lesions are black, and the wood looks corky and misshapen. And if you see this in the field in fall, it looks like someone just burned your vine. It looks really bad, and they really don't stay too long over there. We also found lesions on the berries, and here we have a progeny growing side by side, a resistant progeny here, and one of the susceptible individuals. This is all natural infection. Early in the season, you see these spots in the berries, and late in the season, the infected berries, you see here the rachis, he gets dry, while this resistant vine is completely healthy. So we score these 0 to 1, just presence or absence of the symptoms. The distribution of the phenotypes in these three populations was very promising, because if you look here, it looks like, this is 0. Here you have a key of the phenotypes, 0, 1, 2, and 3. And it looks like in this population that half of the progeny is infected and half of them it's okay, between 0 and 1. In the rhizome by cinerea, we had about 1/4 of the progeny that was infected. And in the rhizome by Illinois, which has even a smaller proportion of vine infected. When we look at the clusters, we have less, the samples are smaller, because basically we have segregation for the male flowers, so not all of the vines bear a cluster. And we also have, in the Chardonnay by cinerea cross, we have a higher proportion of infected vines than in the rhizome by cinerea cross. So we map these cane lesions. We do the standard QTL mapping with the maps that I showed before. And we were really happy to see this huge peak here in chromosome 15 for the in the male map. As explained before, you have a female map and a male map. This is from Vitis cinerea, which was the common parent for the three half-shift populations. In the next population, the rhizome by cinerea population, we found again the QTL in chromosome 15 and another QTL in chromosome 7. And for the third family, rhizome by Illinois, we found the QTL from Vitis cinerea, a QTL from rhizome, and this other QTL, which we think that is coming from Vitis rupestris, which is the grandparent. The allele effect for the most significant marker is the standard dominant allele effect. If you have the resistant allele, the vines have a score of 1, which is just a small lesions. And if you have the susceptible allele, the vines have a score around 3, which is this corky and very bad misshapen wood. In the crosses with rhizome or by cinerea, we also have if you have the resistant allele from rhizome or for cinerea, the vines get just a score 1, which is small lesions. And if they inherit the susceptible allele from rhizome and the susceptible allele from cinerea, you get this bad situation of a score of 3. The third locus from Illinois 547 had a minor effect and explained less than 5% of the phenotypic variation. So we like to put names on this, and in grapevines we use the name of the pathogen. So the dominant locus from Vitis cinerea is called RDA1, and 2 dominant loci in the Vitis cinerea by rhizome cross with the RD1 and RD2 locus. So then we map the lesions on the berries too, and we found that they co-segregate with the cane lesion. So in blue is the interval for the cane lesion, and in red the interval for the berry. And we found that they map. So here we have 2 genetic maps, the rhizome by cinerea and the Chardonnay by cinerea genetic maps. The physical positions correlate well, and the berry lesion and the cane lesion map to almost the same region. So we were able to narrow down this to 370 kilobase pairs for the Vitis cinerea locus. And we look at the annotation of the genome, and we found that here there is a cluster for disease-resistant protein analogs, the NBS-LRR genes, which is really nice candidate genes for a single dominant resistance. So we wanted to do a little bit more, and we wanted to do a EQTL analysis for this. We wanted to try to narrow it down to less candidates, but of course we didn't have the money to do an EQTL for all the samples. But given the high load that we saw before, we thought that maybe if we select a specific progeny, we can get still something that is significant. So what we did was to select 12 resistant and 12 susceptible progeny. Because we already have the genetic maps, we maximized the recombination around the RD1 locus. This is what we selected. We infected shoots in the field with Diaporthe ampelina overnight. We did one pool RNA-seq library, and we look up for the differential expressed transcripts between the group of susceptible and the group of resistant binds. And out of that, we had 16 transcripts that were differential expressed at 0.05. So we use this, these 16 transcripts to do the EQTL mapping, to map these QTLs. And the results that we found that 6 of them were significant, and all of them mapped to the RD1 locus. We found that 2 of the R proteins located in the cluster within the QTL interval were differentially expressed. And for our curiosity, what we found that the transcript for these R gene analogs were more abundant on the susceptible progeny than the resistant progeny. So you might think a little bit for logic that the resistant should have more of these R genes, but we found the opposite way. So that kind of opened more questions, and then we think, oh, maybe this, because the R genes work with the cellular death, so maybe that's opening the gate to the hemibiotrough pathogen. So that's kind of the question that we're trying to solve now. So as a summary for this, we characterized the Phomopsis cane and berry rot in hybrid families. The resistant vines show no symptoms in berries and minimal lesions in canes. We mapped 3 sources of resistance for this disease on North American or hybrid grapes. We localized the locus for one of these QTLs, which co-segregates with the clusters of disease-resistant proteins. And we were able to reduce the number of these analogs to just 2 of them by mapping the transcription level among a subset of the progeny. So for future directions, of course, when you do research, you open more questions than things that you answer. For the maps, for the pipeline for the genetic maps, we were able to create parental maps, but it will be really interesting to create consensus maps and integrated maps. And that will help us to take advantage of the healthy families structures that we have in the Vitis Gen project. This is something that we haven't done yet, but it's something interesting to do. In the case for the biological control of pets with phytoseids, we were able to prove that the genetics of the host is able to provide better habitat for the phytoseids and affect the abundance of the phytoseids. But what we don't know if this has an effect on the prey. We don't know if this is going to be able or not to do biological control. So it will be really, really good to assess the effects of the QTL on the prey and to look for this European red mite and to see if the locus is also associated with the abundance of the prey. And the third question, which is determine the role of the candidate R genes analogs in the susceptible progeny. It will be, what we were thinking is to determine the differential gene expression on susceptible vines after the inoculation to see what type of response is triggering there. So with that, I would like to make my acknowledgements to the groups that helped to support this. I want to highlight here Steve Luce, which is the person that that used to work in the field with with Bruce, and who was the person that spotted the segregation of the Phomopsis resistance. So he was like every winter saying, I see segregation of this trait, I see this trait is segregating, why don't you map it? And finally he took the first score, and it was like, wow, we had huge QTL. I need to phenotype this another season. Jackie did all the RNA-seq analysis, which I didn't describe in extent here, but she did a lot of work on that. Rebecca and Karen, they did all the phenotype assessment for the domatia trait, which is just looking at the microscope and giving a score and counting all these predatory mites. And Kitty Haima, that did the head maps pipeline, she did all the coding for this, and it's basically what we have been using for all the genetics maps and the genetic analysis. And with that, if you have any questions, I'll be happy to answer.